This is a production of Cornell University.
Okay, so my research is focused on the soilborne oomycete pathogen, Phytophthora capsici. I'm going to quickly talk a little bit about some aspects of its life cycle because they contribute to some of the reasons that make it really difficult to control. In this picture on the top left, I'm showing an asexual spore, a sporangium, release 20 to 40 zoospores, which are motile spores. They're biflagellate, and they can swim through water and find new hosts to infect. So the rapid asexual reproduction of this pathogen enables it to really quickly spread in a field under saturated soil conditions. Sexual reproduction, on the other hand, on the bottom left, I'm showing sexual oospores. These spores have a really thick wall and are able to overwinter in New York. They can withstand really harsh environmental conditions, and they can remain dormant in the soil for up to 10 years, making crop rotation an ineffective way to deal with this pathogen. So even though this pathogen is not able to disperse long distances in the air like some other plant pathogens, its ability to spread in water has introduced it to many new farms in New York State in recent years via floodwaters with really heavy precipitation events. So the disease that's caused by Phytophthora capsici is known as Phytophthora blight. It affects a variety of vegetable crops, including solanaceous species, cucurbits, and legumes. It causes different disease symptoms based on the plant part that's infected. So on the left side of the screen, I'm showing what's known as root and crown rot. On the upper left, there's a squash seedling, and on the lower left, I'm showing the symptoms on a pepper seedling. And it also causes a fruit rot, which you can see on the right on snap bean, pepper, and pumpkin. So compared to some other crops, pepper is somewhat unique as a host crop of Phytophthora capsici in the sense that it's the only crop where there are resistant varieties that are commercially available. Plant breeders have discovered resistance in a variety of different peppers from around the world. One of the most important is the serrano pepper on the left that was reported in the 1960s from the Mexican state of Morelos. This pepper is called Criollos de Morelos 334, or CM334 for short. And genes from this pepper landrace, as well as some other peppers, have been introgressed into modern pepper varieties to release resistant hybrids to growers. However, while these resistant varieties generally perform well, it really can depend on what isolate of the pathogen they're infected with. So if you look on the right, I'm showing both the susceptible pepper variety, red knight, it's a bell pepper, and on the bottom, I'm showing the resistant bell pepper, paladin. And when we inoculate these plants with the pathogen isolate 0664-1 on the left, we see that red knight is susceptible and paladin is resistant. However, when we inoculate the same two varieties with the pathogen isolate 1455 on the right, you can see that this isolate is able to kill most of those paladin seedlings, showing how important isolate selection is in determining the resistance level of a particular pepper variety. So we know a lot about the genetics of resistance to this disease in pepper. We know that there's a mixture of major effect genes and minor effect genes. And we also know that there are some genes that have an effect across all isolates of the pathogen and other genes that only confer resistance to particular pathogen isolates. However, the genetics of virulence in the pathogen really aren't understood. We don't know what the genes are that allow some isolates to cause more disease than others, and if there are different genes that are responsible for virulence on different pepper varieties. So knowledge of this could allow us to develop molecular markers that would predict the race of a particular strain in a farmer's field. In other words, a grower could send in, or an extension agent could sample a pathogen isolate from a particular field, run a quick diagnostic test in the lab based on the molecular marker, and determine which pepper varieties would be resistant. So our idea was to perform a genome-wide association study in order to find the genetic variants in Phytophthora capsici associated with virulence on different pepper varieties. So the idea behind this is fairly straightforward. On the right, I'm showing kind of a made-up example where we have 4 strains on the left with a particular nucleotide at one locus in its genome, a G, that are able to kill a pepper variety. And on the right, a different set of strains that have a T at that position and are unable to kill those peppers. A genome-wide association study is just assaying a large panel of individuals for a phenotype of interest and correlating those results to molecular differences in molecular markers across the genome. It's important to note, however, that that particular nucleotide, in this case the GT variant, might not be the actual mutation that's affecting the trait, but just linked or in the same genomic region as the mutation of interest. So in order to do this, we collected 252 isolates of the pathogen. 242 were sampled from 24 farms across New York, shown in the lower left here. And then an additional 10 were sent to us from collaborators in different states. And we genotyped these isolates using genotyping by sequencing, which is a strategy for in parallel sequencing fragments of the genome that are flanked by restriction enzyme cut sites in a population. So using this strategy, we were able to assay this collection of isolates at over 60,000 single nucleotide polymorphism markers. When we look at this genetic data, we found that these 250 isolates were composed of 129 genetically unique genotypes. Many of the isolates collected from the same field turned out to be clones of each other, showing kind of the relevance or the importance of asexual reproduction. And when we build a neighbor joining tree or a phylogenetic tree based on the SNP marker data on the right, we see that isolates are much more closely related to isolates from the same farm than they are to the population at large. So for example, here, each color is a different region of New York, and each symbol is a different field within that region. And if you look in the lower left, you'll see that there's a cluster of purple triangles and then a cluster of purple circles. These represent 2 fields in Western New York that were maybe within 5 or 10 miles of each other. And although the populations on these 2 fields were more closely related to one another than they were to the rest of the isolates, they were still totally genetically differentiated, showing that once this pathogen is introduced to a particular field, most likely via a flooding event or inadvertently brought in with machinery, it tends to stay there and evolve relatively isolated from other populations. So we first used this collection of isolates to perform a genome-wide association study for a couple of other traits of interest. Here I'm showing what are called Manhattan plots. The x-axis shows the position of each SNP, each marker in the genome, and the y-axis shows the negative log 10 p-value for association with the phenotype. So the higher the point is, the more likely that there's a variant in that region that's affecting the phenotype. So on the top, I'm showing the results for mating type. We assayed the isolates for their ability to produce oospores with either A1 or A2 isolates. And we found one region on scaffold 4 where all the A1 isolates were homozygous and all the A2 isolates were heterozygous, seeming to indicate that mating type in Phytophthora capsici is controlled in a way that is analogous to like an XY sex determination system, such that we have. And on the bottom, I'm showing the results for sensitivity to the fungicide mefenoxam. We assayed the isolates for their ability to grow on media that was amended with different concentrations of mefenoxam. And we discovered one locus on scaffold 62 that was highly associated with this trait. And we were able to find a possible candidate gene in this region, which was a ribosome synthesis factor, which seems consistent with biochemical evidence that shows that the way that mefenoxam interferes with oomycetes is by preventing the production of ribosomal RNA. So with these really promising GWAS results, we continued to begin to phenotype 118 of these isolates for their virulence on 12 pepper varieties. So we chose a mixture of commercial hybrids, a susceptible hybrid red knight, a slightly resistant hybrid Aristotle, and an intermediately resistant hybrid paladin. We chose two different sources of resistance that have been used in breeding, including CM334, the serrano pepper that I mentioned earlier, and perennial, which is a hot pepper collected in India. And then we also included 6 lines from a cross between CM334 and the jalapeno variety, early jalapeno, as well as early jalapeno itself. So in all, to inoculate these 12 different pepper varieties with 118 isolates, we did this in 2 reps, and each experimental unit that we evaluated was 6 seedlings. This ended up being around 20,000 pepper seedlings. So it's been a project that's taken a little bit of time. It's been a team effort, but the data is not 100% finished being collected. So I'm going to present just the results from Rep 1, with Rep 2 currently being underway. So when we look at the distributions of virulence for these isolates on the different pepper cultivars, here I'm just showing 6 of the pepper cultivars because we didn't really find many isolates that were able to cause disease on the lines that were from a cross between CM334 by early jalapeno, so I'm not showing them. And on these plots, the x-axis is showing the number of those 6 pepper plants killed, and the y-axis is showing the number of isolates that were able to kill that many pepper plants. So if you look at red knight, for example, on the top left, you'll see that most of our isolates were able to cause high disease on red knight, killing all 6 of those pepper plants, with a smaller proportion able to kill, unable to kill any plants, and very few isolates of intermediate virulence. For Aristotle, we had kind of a 50-50 mixture of isolates that were totally non-pathogenic and isolates that were able to cause high disease in Aristotle. Paladin, the resistant hybrid, most of the isolates were unable to cause disease, and we had a moderate proportion of isolates able to cause a low level of disease and very few able to cause high levels of disease on Paladin. We were surprised to see for early jalapeno a very similar phenotypic distribution compared to Paladin. Surprising to us because we assumed that early jalapeno was susceptible to these isolates. None of the isolates killed any plants of that serrano pepper from Mexico, CM334. But for the other source of resistance, the Indian hot pepper, many of our isolates were able to cause high levels of disease. When we performed the genome-wide association study, we were able to find several regions that were significantly associated with virulence on these different cultivars. If you look on the left side, I'm showing the results for virulence on red knight on the top and the results for virulence in Aristotle on the left. What's interesting to note is that the peaks or the regions that are associated with resistance are the same for these 2 varieties, which is fairly unsurprising given that they're 2 bell pepper varieties. And in this case, they're actually bred by the same seed company, so they should be pretty genetically related. On the right, however, we're showing the results for virulence on red knight on the top and virulence on the jalapeno variety, early jalapeno on the bottom. And you can see that the peaks are totally independent, indicating that the genes that are involved in virulence on red knight are completely independent from the genes in Phytophthora capsici that are involved in virulence on early jalapeno. And although these results only represent half of the data just from Rep 1, I feel pretty confident that they're biologically significant or relevant. The peak SNP for virulence on red knight, for example, was located inside a putative effector protein, which are secreted proteins with a known role in virulence in oomycete species. So with that, I will thank the members of my committee, funding from New York State and the USDA, and many thanks to all the people who made this work possible, especially Garrett Giles, who has done a lot of the work for the pepper virulence phenotyping. Thank you, and if there's time, I'll take any questions.
This has been a production of Cornell University, on the web at cornell.edu.